Thank you. Invisible microfactories. You're all aware, our naked eye, we can see only few things. Few things, it may be from frog egg. It's very minute, we can see through our naked eye. But like yeah, chicken egg and human beings, plant, animals, some animals, we can easily see all those things. But some we could not able to see. With the help of the microscope only, we can see some of the organisms. Which are those microorganisms? What is their role in our universe? What are we will see later. Okay. Okay. Welcome to the microbial world, invisible wonders of life. What it means? This session we discuss the importance of microorganism not only as causative agent of disease. You are all aware the pandemic situation, that is COVID-19 from the past 1.5 years. But what about the previous? We think about our ancestors are 3000 BC, the Egyptian and all those things. They are suffer from so many diseases. Because of evolution and the resistance, now we are in this era, that is in the 21st century. Now we are facing the COVID-19 situation. I will not, because this microorganism not only cause the disease, but in the meantime, it is useful also. The important contribution in the food production, antibiotic manufacturing, and environmental management. Some of our friends in the fellow colleagues are listening in the environmental background. Without the microbes, it is very difficult to decay the things, dead material, whatever the dead material it may be, from the animal source or the plant source or the whatever the source, it is degraded by this microorganism. It's present a brief history of microbiology because it is a versatile group. That is what our close friend Amritesh told. That is why I touched the history of microbiology, how it develops, when it develops, and all those things. Later, I touch 1 or 2 important products that is useful for your research. Based on that, I will tell you something about the endophytes or the wine production and all those things. Here, overview of the microbial world and a discussion of the scope and relevance of microbiology in today's society. That is very important. What is microbiology? Microorganism is usually less than 1 millimeter in diameter, that we call it as microorganisms. 1 millimeter in diameter. We cannot see our naked eye. With the aid of microscope only, we can see those organisms. Microbiology in broad term, when we study the virus, we call it as virology. When we are interested in bacteria, we call it as bacteriology. When we are interested in mycology, we call it fungi, we call it as mycology. When we study the alga, we call it as mycology, and protozoa also comes under this, and other branches that which are the organisms which comes less than 1 millimeter, we call it as microorganism. That is what we call it as microorganism. Now it is believed that only 3% of microorganisms have been discovered. What about the rest? They are waiting for people like you to discover. That is the beauty. We know only 3%. We don't know another 97% of microorganisms. What is the difficulty in that? Because the difficulty is we don't have such a media to isolate and identify all microorganisms. That is the major drawback. Today also we follow the some media, natural media, that is to grow the molds, we go for the PDA media. For the bacteria, we go for nutrient agar media. That is the one that is proposed in the 18th century by Robert Koch. But till now, there is no such media to isolate all the organisms in a single plate or in a single media. That is the beauty of the microorganism. Here, I already told, in our naked eye, we can see the frog egg, chicken egg, and human eye, plants, and something. But we cannot see bacteria and fungus. With the aid of microscope, that is compound microscope, we can see those things. But even smaller than these bacteria and virus, that is bacteria and fungi, we want to see the virus and mycoplasma, we go for the electron microscope observation. With the help of electron microscope only, we can see those organisms and we can identify one. Then we go for the further what is its metabolism, what is its life cycle, and what is the use, or what are the adverse effect of these, and all those things to study. This is the basic for basic to study all those things. This is the simple microscope is invented by Antony van Leeuwenhoek. I will go to the next. The size of the microorganism varies from 0.01 micrometer to 1000 micrometer. How? You see the phase. The size of this page is 0.1 micrometer. Page T4, the size is 0.1 micrometer. That is equal to our COVID-19. That is COVID-19 pathogen. And also the Streptococcus pyogenes, it's 1 micrometer in diameter. Saccharomyces cerevisiae and Bacillus megaterium, the size is 10 micrometer. And Eucaloxicum fischelshoni, this is 100 micrometer. The size is so big, but it is a microorganism. This is the one that is isolated in the sea of Australian, near Australia. Like now, the biggest bacteria is Thiomargentia namibiensis. This is the one, it is isolated near the South African, between Africa and Australian sea region. The size is 1000 micrometer. 1000 micrometer we can see. But this is the beauty of these things. But the macro organism, like for example, mushroom is also comes under this microorganism, Basidiomycetes, but we can easily see those mushroom. It may be butter mushroom or whatever the thing. That also comes under this microorganism. That is the important food, staple food, because it is rich in protein and all those things. That is the beauty of. These are some of the important great guns of microbiology. The first one is Antony van Leeuwenhoek, Robert Koch, Louis Pasteur, Paul Ehrlich, and Alexander Fleming. I will tell about their contributions and their invention, and I move on to the other topics later. First, I will go to the topic, the contributions of Antony van Leeuwenhoek. In the 17th century, actually, he is actually a cloth merchant. Antony van Leeuwenhoek, he was a cloth merchant. His hobby is to grind the lens to check the quality of that thread. He grinded in his lifetime 419 lenses, and he constructed 270 compound microscope. This is the microscope. This is the photograph of that microscope, the size microscope. From this, he magnifies 270 times. 270 times magnifies, and he observed and recorded those pictures and submitted to the Royal Society. Today also we can see whatever the picture he submitted to the Royal Society. It may be cocci, bacilli, streptococci, staphylococci, and vibrio, and all structure it is similar. Today also we can see the same observation. But those days he called the term as animalcules. Now we realize that is a microorganism. That is the contribution of to this. That is actually in the 17th century. 17th century. That is the one that starts the era of microbiology. We have Antony van Leeuwenhoek. We call him as a father of microbiology. Because of his observation and invention of no, he is not invented this compound microscope. Before that, in the 1665, Robert Hooke observed the cells. For all living organisms, cells are very important with the help of crude microscope. But Antony van Leeuwenhoek magnifies 270 from 50 to 270 times magnifies and observed and recorded the pictures. And also he observed the rainwater and also the tooth scabies. And in one of his statement, he told whatever the microorganism is in my mouth is equivalent to the population of our Poland. That is what he stated once in the newspaper. That is the contribution of Antony van Leeuwenhoek. Now we are in the era of electron microscope, but the compound microscope today, our students are using whatever the compound microscope they are using, we can magnify up to 300 times and we can observe this bacteria. But for the virus and all those things, we need electron microscope only. This is the contribution of Antony van Leeuwenhoek, that too in the 17th century. Now we are in the 21st century. In the 17th century, the era starts, that is microorganism. That time he called the microorganisms as animalcules. Now we are calling that as a microorganisms. And the contribution to the microbiology and to the society is more when compared to any other person, that is Louis Pasteur. His contribution is more. He's basically a chemist. He is a professor in the University of Lille in France. France, actually, his contribution is more towards the life science, especially for the microbiology. Some of the people, they consider him as a father of modern microbiology. This era, we call it the golden era of microbiology. In the France, Louis Pasteur was working. At the same time, in the Germany, Robert Koch is working on microbiological aspects. What are the contributions of Louis Pasteur? What are the contributions of Louis Pasteur? He contributed, because in those time, fermentation. In those time, the chemistry ruling the world. And fermentation, the people thought that that is a chemical process. Please mute. Please mute some
06.
Dear participants, please mute. Okay. Fermentation in those days, because you know the importance of chemistry, in those days the people thought that fermentation is a chemical process. Fermentation is a chemical process, but he is the person in the wine production. He clearly demonstrated the world that is not the chemical process because of the microorganism. The carbohydrate is the one, it is converted into alcohol plus carbon dioxide, the help of the microorganism, that is Saccharomyces cerevisiae yeast. The yeast is the one, it shows very clearly the fermentation is not a chemical process, it is a with the help of microorganism, the conversion occurs. Now, from the fermentation, so many pharmaceutical industry and acetic acid and hormones, vitamins, lactic acid, citric acid, and invertase, and so many other products comes through this fermentation. And he is the first person observed and reported the aerobic and anaerobic bacteria, and also he is the one reported facultative anaerobic microorganisms also. That is the great contribution. I will tell you later, after their invention, I will take you to the wine production. That time, what is the reaction, I will tell you later. It is very easy why wine is important and its significance, I will tell you later. And in the same product, you observe the pasteurization. For the honor of Pasteur, we call it as pasteurization, to removal of unwanted microorganisms. What do you mean by pasteurization? Pasteurization is the one, it is nothing but heating the temperature, that is 62.5 degrees Celsius for 30 minutes, the liquid, it removes the unwanted microorganism, not all microorganisms, unwanted microorganism. It is not ability to destroy the endospores containing microorganism. The other, that is the one proposed by Louis Pasteur. That is what we call it as, for the honor of him, we call that technique as pasteurization. Now we are high temperature, short time pasteurization, that is 72 degrees Celsius for 15 seconds. Everyone in the household, when they are pasteurizing the milk, they know the things. And ultra pasteurization in the milk factories, they are using ultra pasteurization. In the ultra pasteurization, 138 degrees Celsius for 2 seconds and sudden cooling. That is 3 types of pasteurization now following. In the 18th century, Pasteur proposed the pasteurization, that is 62.5 degrees Celsius for 30 minutes. This is very important. Why he proposed? Because he proposed this to save the wine industry. Because if you boil in high temperature, it changes the taste and odor and color, because these things are very important in wine. In this wine production only, he come out with the pasteurization and fermentation, and he invented the vaccines for chicken cholera, anthrax, and rabies. If you are it is a diverse group, I will tell you some of the stories here because it is very interesting. Because you know who is the father of vaccination. Now we are all vaccinated for COVID-19. The first vaccine given by Edward Jenner in the 17th century. He is actually a physician. By his observation, 1801 in India also we are vaccinated for smallpox. How he invented this smallpox? Because for the honor of Edward Jenner, Louis Pasteur coined the term vaccination. Vac means cow, vaccination. That is the word is proposed by Louis Pasteur for the honor of Edward Jenner. Father of vaccination is Edward Jenner. Edward Jenner is basically a physician. He is working from 150 miles away from the London. In London, he is working in the area in a village. In that village, in those days, smallpox is a major disease. Died because of smallpox. He observed one milkmaid lady is not suffering from this smallpox, but she suffered from cowpox. He observed 3-4 months and she is not suffering from any smallpox symptom. She is suffering from cowpox because milkmaid, she used to take the milk, whatever it is this thing, it is infected her body. In his observation, one day he called her and he collected the pus from that cowpox cells and he injected to one boy. Boy name is James Phillips. 9-year-old boy. That time there is no such injection. That is so many types of in that time there is no such injections. In a knife, he touches the pus cells and transferred that one to his hand, cross hands, regularly for 1 month. But that boy feels uncomfort and discomfort, feel fever and all those things. After 3-4 months, surprisingly, he will not affected by this smallpox. That is the first invention of vaccination by Edward Jenner, by social. By this, and later that is the one, it is extended all over the world. From here to India, the first vaccinated against smallpox is 1801, but it is invented in 1976. That is in England. Next, anthrax for this thing, cow, and the rabies. Rabies is the biggest name given for this Louis Pasteur invention of this rabies vaccine. Today also, if the person is infected by the rabies, there is no cure. If the disease occurs, this rabies disease occurs, there is no cure even today. But prophylaxis is there. That prophylaxis we call it as vaccination. That invention is also one big story is there. In those days, Robert Koch is succeeding in culturing the bacteria and all those things. The same thing, the rabies is a major problem. The mad dog bite, it may be king or servant or soldier, whatever it may be. If it bites, the death is confirmed. In those bited person, the villagers are banned to that village. They should leave outskirts of this village in those days, and the death is confirmed. And also the other name for this rabies is hydrophobia. If they hear the sound of water, they are very aggressive. But rabies, how to Robert Koch students, they are running behind the mad dog, and if the same mad dog bite this person, definitely they will. There is no cure for in those days. But they take this. They captured this mad dog and collected the saliva, and they try to isolate it in the lab. They fail to isolate these things because in those days they don't know this is virus. Rhabdovirus is the causal organism of rabies. But in those days, they don't know. But try to isolate in the laboratory, but they fail to isolate because most of the viruses are obligate parasites. It requires host. They injected this saliva to the rabbit. After sacrificing this rabbit, they collected the spine and brain from the rabbit. They make the powders and they preserve. That's it. But one day, Joseph, Mr. Mother, came to Louis Pasteur and she begged like anything, please, Professor, you save my son, because anyhow he is bitten by the dog, mad dog. Anyhow, death is confirmed. You do anything to him, and you please survive from this rabies. But Louis Pasteur is not a physician. He don't have a rights to give anything to the patient. in those days. But in the same time, from England, Joseph Lister, he is basically a physician to help Louis Pasteur. He was in that lab. He take the powder and mix it with the glycerin and injected that boy. That is, his name is Joseph Mister. Joseph Mister, 16-year-old boy. They injected this thing for 24 days. After 24 days, fortunately, this boy survives. He escaped from the rabies disease. That invention gives a name and fame to the Louis Pasteur. All over the world, the people came and approached the Louis Pasteur for the vaccine. This is rabies. Whatever the revenue is earned from this, the first institute by name started in the world is Pasteur Institute in Paris. Pasteur Institute in Paris, by name, by person name, started the institute, that is Pasteur Institute, that is situated in Paris today. Also, the Pasteur Institute is there. In the Pasteur Institute, in the top dome, Joseph Mister photo is there. Every day, Louis Pasteur used to sit in that below that dome and see this Joseph Mister photo. When you visit this, anyhow, if you get that opportunity, you please visit Louis Pasteur Institute and see those things, whatever is aseptic water and swan neck flask and all those things. It is very fantastically they arranged like anything. That is his contribution of Louis Pasteur. And Pasteur discovered existence of life in absence of oxygen. I told this is aerobic and anaerobic, and also I told the facultative anaerobes because facultative anaerobes means it survives in presence of oxygen or in absence of oxygen also. That microorganism we call it as facultative anaerobes. Pasteur finding in those days the spontaneous generation. He is the person disproved the spontaneous generation. This is in those days the people thought that organisms are emerges from dead and decay material. But he is the person in the swan neck flask experiment. He clearly told for every organism ancestors are very important. The microbes would only appear in broth if they were allowed in with the air. He clearly showed that even for microbes, life comes only from life. Microscopic beings must come into the world from parents similar to themselves. That is how he disproved the spontaneous generation theory in the 18th century. Till that, the people thought that from the dead and decay material, the organism rises themselves. But he clearly showed for every organism, the ancestors are very important, parents are very important. That is his great contribution to the society. And I told he is the one, and another, okay, the first vaccine is invented by him is the chicken cholera vaccine. That is also accidentally invented by his lab. He given the culture to one of his students. His job is to culture that inoculum and inject it to the chicken and see the symptom of this thing. When he went for the vacation, after 1 month he came back and he tried to inoculate the whatever he cultured, the same culture he inoculated to the chicken. He failed to get the cholera, chicken cholera. He is very feared and reported to the Louis Pasteur. And Louis Pasteur thought that, this may be because it is attenuated, it loses its virulence. That is the first culture lab. This thing is the one is used as a vaccine. That is flu cholera or chicken cholera. Because of this invention only, he is succeeded in rabies vaccine. That is his great contribution, the rabies vaccine. Today also we are following the same thing. And Louis Pasteur investigated on fimbrial disease of silkworm and come up with solution by suggesting the farmer to use healthy disease-free pillars for breeding stock. These are some of his inventions to the society. But in those days, for his invention, so many countries are inviting him to their countries. But he clearly rejected all those things. He is not even patented whatever the success. He was not patented. That is, those things are for the society. Whatever he touches, that is his great thing. He succeeded in overcoming all those problems. That is why most of the people, Louis Pasteur, we consider him as a father of modern microbiology. Next thing is Robert Koch. Today, to establish any disease, we follow the Koch postulate. That is the great contribution to the world today. Today, whatever the disease establishment, we should follow the Koch postulate. That is the one. It is proposed by Koch for anthrax. And what do you mean by this Koch postulate? I will tell you here. For LD and disease, you can see the difference, LD and disease. In the disease, you should isolate the organism from the disease animal, or it may be plant, and you cultured in the lab and identified whatever you cultured in the lab, the same thing you transfer to the LD one. Whatever the seconds, whatever the culture you are isolated, you culture transfer to the LD one. After the LD, that you see the same symptom what you observed in the previous one. And then again you isolate the disease material and identify. This is how you should prove the Koch postulate for any disease. Today, in the recently COVID-19, that is coronavirus, we identified based on this Koch postulate today to establish any new disease. This is must. This is the biggest contribution of Robert Koch. Is basically a physician from Germany. He is a doctor, basically a physician doctor. He worked on this Koch postulate to establishing disease. This is very much necessary. And he made the first micrograph of bacteria. And some of our botany and microbiologist teachers, professors, they know the heat fixing important. That is the one. It is proposed by Koch only, and he is the one identified 3 organisms, that is anthrax, Mycobacterium tuberculosis, and Vibrio cholerae. For to identify the Vibrio cholerae causing organism, he visited Calcutta, India, in the 1890 or 1892, Calcutta. Koch was the first observed the healthy human beings could be carrier of causative bacteria, and it's responsible for spreading the infectious disease. That is why these are some of the contributions of Koch, Robert Koch, and he is the one who developed the culture media, that is nutrient agar media, the same media today we are using. One of his students proposed agar, that is Walter Hazy, one of his students, for as a solidifying agent. It will not contain any nutrients. That is his contribution of Louis Pasteur. Next, Paul Ehrlich. Paul Ehrlich we consider him as a father of chemotherapy. Chemo means chemicals, you know. Most of the disease, whatever it may be, fever, cough, whatever are pain, these things, we are using drugs. The first time this Paul Ehrlich used screening so many compounds and stains against syphilis. But in the compound 606, he was succeeded. Now we call that compound 606 as salvarsan, and he called that as a magic bullet. Why he called that as a magic bullet? This is because it target only to the target pest, not the host. The therapeutic index is more. Today also, whatever the drug we are using, it should reach to the target, not the host. That is where the therapeutic index is more. The side effect is less. That is why most of today's drug is more side effect rather than the therapeutic index. Here the therapeutic index is more. That is why he called as magic bullet, able to specifically destroy tumor cells and microorganisms. And Ehrlich proposed the theory of immunity, in which antibodies were responsible for immunity. If you have a very good immunity, you can overcome with this pandemic very easily. Otherwise, it's very difficult, because that is why the government and the doctors are always telling whatever the people are suffering from any metabolic disorders, maybe immunity, that is autoimmune disease, or the diabetes and all those things, they are very much prone to this COVID-19 because they are very less immunity when compared to other. He developed a staining procedure to identify tuberculosis bacilli. This is also very important contribution in those days, because Christian Graham is the one who developed the staining technique to identify gram positive and gram negative organisms. but they failed to isolate the Mycobacterium tuberculosis. Today also we are following a different procedure to identify the Mycobacterium tuberculosis. Today also there is Mycobacterium tuberculosis strain identified by Robert Koch in the 17th century. Today also there is a no, because it develops very quickly develops this resistance. To overcome, we call it as multi-drug resistance. Today also we are suffering, and now government of India, we are following the DOT therapy for Mycobacterium tuberculosis, those who are suffering from tuberculosis. Today also the say that is to stain those things is the person developed the staining technique to identify the tuberculosis. And Alexander Fleming, his best known discoveries are the discovery of the enzyme lysozyme and antibiotic substance, that is penicillin, from the mold Penicillium notatum in 1928. This is also one of the accidentally got that stain, that is Penicillium notatum, out because Alexander Fleming cultured the Staphylococcus and he went for the vacation. After vacation, he came and he observed that plate. In that plate, he observed one mold. Around the mold, he observed some zone of inhibition. What is that zone? Oh, it is not allowing, this mold is not allowing this bacteria to grow. That is his invention. That is why he is the father of antibiotic. That is the first invention in 1928, penicillin from Penicillium notatum. The Florian chain took the culture of this Penicillium notatum culture from Alexander Fleming lab and purified, and penicillin yield was very less. Now today we are using Penicillium chrysogenum, but penicillium, why this penicillin in those days is very important, because in the Second World War so many soldiers are died because of the wound infection. To overcome this, penicillium played a very important role in the Second World War. Penicillin, to get that penicillin, most of the they saved the life because of this penicillin. 1928, that is Alexander Fleming, Florian chain got the Nobel Prize of his discovery of penicillin. Saved millions of lives with the discovery of penicillin. Even now, today also, for most of the gram-positive, penicillin is very effective, and therapeutic index is more when compared to any other antibiotic, because it targets only the cell wall of bacteria. Cell wall containing only peptidoglycan present only in the bacterial cell wall. That is why the therapeutic index is more. But some people are allergic to this penicillin. The second discovery is from the Septomycin. I'll tell you later. I'm boring.
No, sir,
no boring. It's very interesting.
Proceed, sir. Thank you. Because I don't want to take you to the research aspect. I will touch one or two research aspects, what you can do in future and all those things. Before, because of that, I am giving only the introduction. I told already important invention. Walter Asay used agar as a solidifying agent because it will not contain any nutrients. That is his greatness. What about the beauty of this agar? When you heat it above 50 degree, it is in a liquid form. It comes below 50 degree, it is in a solid form. That is the beauty of the agar. It is the one, actually, Walter Asay's wife, actually, she is the one suggested, in those days, people are using gelatin. Gelatin is very quickly degraded by this microorganism. She suggested, why don't you use the agar? The same thing is also, today also we are using the agar. It is, the product comes from the algae only. Algae is nothing but microorganism. Next, Richard Petrie, whatever the Petrie plates we are using in the laboratory, that is what invented by Richard Petrie. The size is varies, what type of Petri dish we are using. Usually for microbiology, we are using 10 centimeter, that is diameter size. We are using the Petri plates in the laboratory to culture most of the bacteria and molds. And Christian Graham is the one proposed the staining technique. Today also, for the basic thing, we go for the staining, that is gram positive and gram negative staining. Most of the gram negative bacteria are pathogenic when compared to gram positive bacteria. Today also, most of the gram negative bacteria are dangerous when compared to gram positive bacteria. And Raymond Sobodox is the one developed the culture for the yeast and mold. And Ivanovsky, his contribution tobacco mosaic virus, the invention, the rabies vaccine invented in 1885 something, but actually the virus TMV, the first virus, tobacco mosaic virus, identified by Benzeric and Ivanovsky, because they are filtering this to remove the bacteria, but it is escaped from that filter. Then they crystallized and identified that organism is tobacco mosaic virus that is started in the 19th century. Now we are in the 21st century. Now we are realizing the importance of that virus, how it pandemic and all those things. And Salman Waxman is the one discovered the second antibiotic, that is Septomycin. After Alexander Fleming, the whole world is very much interested to search for the new antibiotic from the microbial source. This Salman Waxman screened 6,000 cultures from soil. In that, he identified one that is Streptomyces graecis, that is actinomycetes from the soil. Today also for the so many antibiotics, that is the base. One is natural, and we divided the antibiotic into 3 groups. One is natural antibiotic, semi-synthetic antibiotic, and synthetic antibiotic. Most of the semi-synthetic antibiotics are coming from this source, that is Streptomyces graecis strain only. It may be tetracycline, ciprofloxacin, so many other drugs. Now we are in the 4th generation of antibiotic. Why it is a continuous process to invent this antibiotic? Because they develop the resistance. I told already Mycobacterium tuberculosis. Today also there is no, it is a developed resistance. We are continually using new antibiotics. That is why we are using, now we are in the 4th generation of antibiotic. Now it is a continuous process of discovering the new molecules to overcome the problems of these microorganisms. What is the scope of microbiology and microorganism? Microorganisms were the first living organism that is on the planet. Live everywhere life is possible. You are all sitting in the chairs. They are also microorganism. Air, water, everywhere microorganism is there. They are more numerous than any other kind of organism, and probably constitute the largest component of Earth biomass, because we know only 3% of microorganisms. We don't know the another 97% of microorganisms. That is the beauty. That is why the role of, because some are useful, some are harmful. In our intestine also some E. coli is there and probiotics is there. Some important microorganism is symbiotically associated and it is helpful for the human and also the plant system. Some are harmful. Like that, in the symbiotic association, we are all living. The entire ecosystem depends on the activity of microorganisms. The organism influence human society in countless ways. Microorganism is an important in medicine. I told one way it causes the disease. In another way, the same microorganism, we are using it for the antibiotic and the production of hormones and also the production of insulin and all those things. In the agriculture, in the one way it causes the disease, and another way is the same microorganism, we are using it as a biofertilizers and also the biopesticides. In the food science, some way it spoils the food, and say in the other hand, it helpful for the probiotics production of so many dairy products is come from this microorganism. Ecology balances the ecosystem, ecology, genetics, biochemistry, and other fields. I will touch one or two, and I will tell you one production of wine, and I will take you to the other field where the unexplored area of microbiology. The next is in the agriculture. I will tell you the human, only one pathogen, how destroy the economy. In Irish famine is the one. The late blight of potato, causal organism, now we know Phytophthora infestans. That is the one, it is identified by D. Barry. But in 1845 to 1852, the major food for the Ireland is potato, staple food, because of the mass starvation. 1.2 million people died because of starvation, and 2 million people migrated from this island to US. the only one pathogen, that is Phytophthora infestans. But in those days, they don't know which is the causal organism. That is the Irish famine. One microorganism devastated the whole country, and also the economies comes down. 2 million people migrated from to US, and 1.2 million people died because of starvation. In Ceylon, 1865, that is leaf rust of coffee caused by Emilia vastadix. Because of that organism, the whole Ceylon, whatever now we call it as Sri Lanka, they switch over from coffee plantation to the tea plantation. That is the beauty of this single microorganism. The Bengal famine, 1943, that is the brown rust of paddy, causal organism is Helminthosporium. Because of that, so many people are died in Kolkata and West Bengal because of this famine in 1943. And also in the meantime, it is the one, it helpful for the soil fertility, and such as microbial degradation of organic matter and soil nutrient transformation. It is very much useful. And Azotobacter, Rhizobium, Azospirillum, Cyanobacterium, phosphate solubilizing microorganisms. These are the one we used as a biofertilizers. And Bacillus, Pseudomonas, and Trichoderma. These are the one we consider this as a biopesticide to kill the pathogen. We are using this microorganism. This is the beauty of the microbes. Next, in the food industry, some of you are in the morning, some of you ate the bread, idli, dosa. These are all microbial product, and these are all we call it as probiotics. These are very useful. We knowingly or unknowingly in our life, day to day, we are closely associated microorganism, and we are using this microorganism for our use. Bread, idli, yogurt, cheese, carry, buttermilk, sour cream, acidophilus milk, and dahi. So most of the dairy products are come from this source only, and alcohol and so many other products, food products, and whatever today we are using, that is gobi manchurian and all those things, they are using vinegar. Vinegar is nothing but acetic acid. Where you are getting this acetic acid? This is also a fermented product. Like that, so many products are coming from the microorganisms. Like in the industry, we are go for the fermentation process for antibiotic or in the chemical preparation, human growth hormones, human insulin, and hormone. Human insulin, you know, before we are using bovine insulin and all those things. Now we are using human insulin. How? Because of the genetic engineering, we isolated that insulin and injected that insulin into the E. coli. From that E. coli, we are getting the insulin. It is similar to human insulin. Yeah, these are all some of the products, industrial products. Not only that, acetic acid, lactic acid, citric acid, ethanol, lipase, invertase, and single cell protein. These are all from the microbial source only. We are these are useful in our day-to-day life. We are knowingly, unknowingly, we are using this. Next, I am moving another product. I am already okay. Production. One important product is wine, because in the 18th century, the Antony van Leeuwenhoek observed and Louis Pasteur showed the fermentation process, microbial process. But 3000 BC, Egypt and Persia, two areas of the Fertile Crescent, the most probably witnessed the birth of fermented spirit. This is what 3000 BC only. People are using this wine, but that time the people, they don't know that is from the microbial product. The study of these and wine making, the separate branches there in the science, that is enology. Wine production or science of enology, we call that as enology. Today also, this is very important subject in Europe. People enjoy drinking wine for thousands of years, ever since it is the origin of Mesopotamia, Egypt, and Neolithic period. Wine is the common usage defined as a product of the normal alcoholic fermentation of the juice as a sound of grape juice, that is sound ripe grapes. Any food, not only a grape, any food containing carbohydrate, it is associated with Saccharomyces cerevisiae, you will get the product that is alcohol plus carbon dioxide. That is a very good. Why I will tell you the I will tell you how the production of wine we can make in our house only. Why it is very important? Is it important? Yes, it is very important because it may be grape or guava or cyzicium, whatever the fruit or cherry containing the carbohydrates, we can use it for the production of wine. Because of 2024 is the grape, and very different strains, that is different varieties we are using and different strains we are using. Today also the French and German wines are very much superior than any other wine, because in the grape, whatever the nutrient content, what type of water, what type of strain, or in which area we are growing, in the soil, what are the mineral content, these are all play a very important role in the wine production. And in the wine, which are the one is very important here in the wine, the taste, bitterness, color, and preservation and oxidation, these are very important. Today also taste and bitterness and color is very important. How the colors are varying, what type of fruit we are using. For example, if you are using white grape, you will get white wine. If you are using the black grape, you are getting the red wine, because that color comes from the skin of the grape only. That is vintage vinifera. It contains phenolic compounds. One is flavonoids and non-flavonoids. What are these flavonoids and non-flavonoids are very important, because in some of the studies, it has an anti-oxidative properties of red wine. It against in vitro LDL oxidation. This is in fatty acid metabolism. This is very important to attribute mainly for the presence of phenolic compound. Wine polyphenols have been the scavenging peroxynitrate that exhibit antioxidant, anti-inflammatory, and anti-anthrogenic effect. This is very important. That is the property of wine. Recently, the stilbene and resveratrol with respect to their cardioprotective effect, because this is the one it saves from the that is heart problem, that is heart attack and all those things. Not that is where stilbene and resveratrol, these two compounds are very important to overcome this problem. And also here one of the reports says cyanidine, another phenolic compound. This is the one possesses endothelium dependent relaxing activity in blood vessels in vitro. Blood vessels, you know, when blood blocks occur, the heart attack occurs, and you should go for surgery and all those things. To overcome, this compound is very important to relaxing the blood vessels. And resveratrol has been found to increase the activity of anti-aging gene. Resveratrol is not only protect from the cancer or the heart attack or the kidney or brain, it also useful for the anti-aging gene. If resveratrol is the one, it is responsible for anti-aging gene. That means if you are aging also, you are look like a young. That is the compound is present in that. It helpful for the anti-aging gene. Why? I told very clearly in those days, the people thought that this process is a chemical process. But this carbohydrate C6H12O6, in the presence of Saccharomyces cerevisiae variety ellipsoides, it converted into alcohol plus carbon dioxide. This is what it showed. This is a microbiological process. The conversion of grape juice into a alcohol product plus carbon dioxide. In those days, the Egyptians and all those things, they use as a refreshing agent. When the guest comes, they serve this wine to refresh them. Like today we are using coffee and tea and all those things. Like in those days, it is a prestigious issue if you offer a wine in Egypt and all those things. How to prepare this wine? Simply, I will go through you go to the market and you select the grapes. Maybe if you are interested in white wine, you select the white grapes, or if you are interested in red wine, you select black grapes. You can take 5 kg and wash thoroughly and remove the damaged and unwanted stem and all remove thoroughly. In the 5 kg, half of 2.5 kg you squeeze and you store it into a glass container. Don't store it in the metals. You preferably use glass container, because when the reaction occurs, the corrosion occurs. That is why you should use the glass containers. You fill it in the glass containers and plug it with cotton plug. Why I am telling this cotton plug? Because grape itself associated with Saccharomyces cerevisiae yeast. If you are really interested to some good volume of alcohol, you add baker's yeast, that is Saccharomyces baker's yeast, to that container, and you plug it with cotton plug. Why I am telling this? Aeration is very important for initial process. In the initial, Saccharomyces cerevisiae multiplication occurs, and it increases the biomass of Saccharomyces cerevisiae. In the aerobic stage, the Saccharomyces cerevisiae multiplication, growth occurs, multiplication occurs, the biomass it increases like anything. It utilizes carbohydrate, and the biomass of Saccharomyces cerevisiae increases in the glass vessels. In the alternate days, in the 5 kg, your 100 grams of sugar you mix thoroughly and you pour it into the plug, and again you close it with cotton plug. After 14 days, alternate days, you add the sugar 100 grams. Why? Because if you are increasing the carbohydrate, the metabolism of this Saccharomyces cerevisiae also increases, and its multiplication occurs. The mass production of Saccharomyces cerevisiae increases. Because of that, we are alternate days we are adding the sugars. After 15 days, you completely close the container. You switch over to anaerobic condition. When you switch to anaerobic condition, the Saccharomyces cerevisiae is the one, it produces alcohol. When it produces alcohol, it releases carbon dioxide. When it produces alcohol, you keep it another 14 days. 14 days for aeration, and 14 days you keep it in anaerobic condition. In 14 days, the Saccharomyces cerevisiae releases alcohol. The maximum alcohol percentage in the wine you can expect only in maximum 12 to 13. In the home, we can expect only 8 to 9%. After completion of 28 days, you filter the grape and remove the pulp and all those things. Whatever the filtrate, you go for pasteurization. Why I told? Because in this process only, Pasteur observed the facultative anaerobes, Saccharomyces cerevisiae, and to keep it for a long time, you after filter, the filtrate should be pasteurized. That is very important. 62.5 degrees Celsius for 30 minutes. You should not exceed the heat. If you exceed the heat, the aroma, taste, color vary. That is why that is the ideal temperature to for pasteurization. Ideal temperature is 62.5 degrees Celsius for 30 minutes. After pasteurization, you preserve this wine for a long time. If you preserve the wine for a long time, the value of the grape and also the rate increases because of the taste, aroma, smoothness, and all those things comes from this wine. This is from the one product. This is very useful nowadays. So many people are suffering from heart attack and all those things. And this is resveratrol is the one, it is present in red wine. This is very important. That is why I told very clearly the production of wine. It is very simple. You can, most of the couriers in our area, most of the poor people, they are preparing this wine from different berries and fruits. If you visit Kodagu, you can take this ready wine, you can take and you taste it. Not in a large volume, in a small volume. I am not insist you to take, but I am, what are the significance of this wine, and the production is very easy and its importance I told. That's it. Okay, I will next, some more time is there. I will quickly move on to the unexplored areas of in our lab, that is endophytes. What do you mean by endophytes? Dave Barry introduced the term endophytes are those microbes that reside inside the healthy tissue of plant without causing any apparent disease. That is what we call it as endophyte. Endophyte is a symbiotic association. Endophytic microbiome forms a significant part of microbial diversity. And also endophytes comprises a large hidden component of fungal diversity, biodiversity. Those they want to do and explore this thing. I'm telling you, simply I took this to those who are interested in research to explore this microorganism. I will tell you later how to isolate endophyte, how to identify the biological molecule, and how to check the biological activity and all those things. I will glance very quickly this thing. First anti-cancerous drug is from this endophyte, that is Taxol. Taxol, the first anti-cancerous drug is from endophytes only. That is produced from Taxomyces antriani, endophytic fungus from Taxus brevifolia. An unexplored and untapped source of novel bioactive metabolites we can expect from these endophytes. Diverse biological activity due to their unprecedented structural diversity. That is very important. That it shows. This is how the diversity of fungal community. You are all know. That is the pathogen, leaf pathogen, root pathogen, and endocytosis of microbes via root air, and uptake of organic carbon, nitrogen, phosphorus from soil organic matter, and also increases and reduces the nutrient availability, phosphorus solubilizing bacteria, siderophores, nitrogen fixing bacteria, mycorrhiza, plant growth promoting rhizobacteria and fungi, carbon root exudate, attract heterotrophic microbes, like that. So many symbiotic association, and some are pathogens, that is leaf pathogen, root pathogens, and this is epiphyte microbes. It is on the surface of this plant system. We can see the plant microbial interaction in this plant. But endophytic fungi inhabit almost all variety of plant species. The diversity of fungal endophyte mainly depends on species and geographical area. Plants with ethnobotanical history from important criteria towards screening. It may lead to the isolated endophyte with a potent activity, and few of the scientists' reports suggest that the plant growing in the endemic that is large diverse group of endophytes with activity. Endophytes, that is what the role of endophytes. How it differs from the disease. For the disease, three factors are very important. One is susceptible host, virulent pathogen, and congenial environment. When these three coincide, then only you can expect the disease. You have a big question. You know why this COVID-19 is not affecting me, because I am not wearing any mask and all those things, because their immune system is so aggressive and his body is resistant to that pathogen. That is very important. Here, when three factors coincide, then only we can expect the disease. Otherwise, no. Here, the entry of endophytes is through the seeds, wounds, and natural opening. Maybe lenticels, stomata, and this thing, and balanced antagonism hypothesis occurs these plants and endophytism. That is a balanced antagonism. We call that as an endophyte. It is residing inside the cell, and it is helpful for the growth of the plant, and also it protects from the adverse environment. It increases the yield of the product and all those things. It will not damage the host. That is where the endophyte play a very important role in the plant system. How to isolate this endophyte, how to screen this endophyte. One is screening the diversity of this endophyte, how it is different from others and all those things. What are the importance of these endophytes? One is we can use it as antimicrobial agent, plant growth promoters, anti-cancerous, anti-diabetic, anti-inflammatory, agro-important metabolites, and anti-diabetic. This is how we can expect from this novel endophytes and also the novel metabolites. We expect this type of compounds, novel compounds, but we don't know whether it's a novel or it's already proved. When we screening, then only we come to know those things. Application in agriculture. Here, the symbiotic association is co-evolution of endophytes with host plant, changes in microbial diversity in their genotype, stage of growth, physiology, plant organ, and other ecological parameters. Why I am telling this thing? The same plant, you cannot expect the same endophytes from the same plant, because it changes its from genotype, stage of growth, physiology, plant organ, and ecological parameters. If whatever the endophytes I am isolating from, for example, it may be from paddy plant here in Mysore, it will not be the same in Tamil Nadu or Kodagu or Mandya or Bangalore, like that. it varies from place to place. That is where the symbiotic, that is co-evolution of endophytes play a very important role. This endophyte brings a change in cellular and molecular level, which is highly complex. We don't know what happened in those symbiotic association. The intimate relationship occurred between endophyte and host plant is a positive selection process from the confers beneficial for successful propagation and survival. They occur plant with negative inherence of biotic and abiotic factors. These interactions are not only helpful for the plant growth, plant health, development and production, but also soil quality, and which could stabilize adverse ecological imbalance due to the various anthropological activities. The positive effect of significantly increased plant biomass, dry matter yield, and grain yield would also income for agronomically with sustainability. This is what the association of plant and endophytes. Direct mechanism of plant growth promotions, production of phytohormones. You are all aware of auxins, gibberellic acid, ascorbic abscisic acid, and cytokinins. These are the one it helpful for the production of phytohormones. And ATD, 1 amino cyclopropane, 1 carboxylate deaminase activity increases the plant growth, and it lowers the production of ethylene. And also it helpful for the macro and micronutrients. It may be carbon, nitrogen, hydrogen, and molybdenum, zinc, sulfur, and all those things. It increases the production of macro and micronutrients. Abiotic stress, it overcome from salinity, high temperature, stress, and all those things to overcome abiotic stress, where these endophytes are very helpful. And phytoremediation, it is the one, it avoids the toxic metal to enter the plant system. That is why we call it as phytoremediation. Indirect mechanism of plant growth promotion, antagonism, you are all know. The microorganism is the one, it is suppress or inhibit the pathogenic microorganism. For example, trichoderma, or it may be bacillus. That is what we are using as a biocontrol agent. Trichoderma, pseudomonas, bacillus subtilis. These are the one we use as a biocontrol agent. It is nothing but antagonism. Induces systemic resistance along with the immunity in the plant growth promoting system. And also application in medicine, it is I told already, the antimicrobial activity, it is a continuous process to overcome the resistance, because we want to kill the pathogenic microorganism, but pathogenic microorganism is also survives in our environment, in the ecosystem, to balance our ecological system. They resist to the existing antibiotic. To overcome, again, we are searching for the newer antibiotic. And anti-cancerous agent, anti-helminthic agent, antioxidant to increases the phenol content in the plant system. These are some of the applications of these endophytes. Criteria, what are the criteria to select the plant? Plant should be unique environmental settings, especially with unusual biology, novel strategy for survival. And also plants that have an ethnomedicinal history. Ethnobotanical history, you are all know tribal paper. They are using to cure disease. They are using some plants. Those plants we can we can go and screen for those plants. We can isolate the novel endophytes. And also plants that are endemic have an unusual longevity. Endangered species occupied a land mass more likely to harbor endophytes with active natural products than others. Plant growing in areas with great biodiversity, we have a rich Western Ghats diversity. Also have a prospecting of housing endophyte with great diversity. Those who are interested in research, I am only touching in another 5-10 minutes. I wind up. These plants, if you select, you can identify the novel. That is the sample site of epiphytic orchid, natural habitat of Dendrobium herbaceum and Trastoxy, and we cultured on water agar media. Whatever the culture we got, we transfer into a media containing PDA to get the pure cultures. These are some of the medicinal plants as a host for endophytes. Ficus pumila, Mirabilis jalapa, Annona swamosa, and Coffee arabica, Cryptolopsis buchanani, Combatium lantifolium, and Croton bonplandianus, Andreatum philippines, Trastoxy, like that. These are some of the plants we use it for the isolation. How to isolate these endophytes? How to remove the epiphytes? Because first and foremost, we can go for based on the important. If root is important as a medicinal, we go for the isolation of endophytes from root. If stem is important in the medicinal thing, we go for the stem. Or if leaf is very much important as a medicinal aspect, we go for leaf. Any part you take, that the healthy one, and you remove the damaged and infected one, wash thoroughly with the running tap water. After that, 75% of ethyl alcohol for 1 minute and 4% of sodium hypochlorite for 4 minutes, 75% of ethyl alcohol for 1 minute. After that, rinse with sterile distilled water, and the laminar flow, you cut it into a small pieces and place on the water agar media. After that, yes, after 7 days, if you observe any colonies, you transfer into a PDA containing PDA media. From that, again, it shows any activity. For the identification of the fungus is very easy. With the help of compound microscope, with standard manuals, we identified the generic level. If you want, first you identify the generic level. If that fungus showed very good biological activity, then you go for the molecular identification. I heard yesterday, you heard the molecular approach, how to identify the molecular technique to identify the bacteria. The similar way for fungus also, isolation of DNA, PCR amplification, sequencing, submitting that one, and blast it, and we get the species name, and we try to plot the phylogenetic tree and identify the fungus. The same way here also, after this thing, we transfer into the media growth and identified the phylogenetic tree. Time that's it.